Welcome back. In this high-yield explainer, we are jumping straight into the lab diagnosis and prevention of diphtheria. We're going to focus on exactly what you need to know to confirm a case, stop it from spreading, and of course, absolutely ace your exams.
Okay, before we even look at a single lab test, let's get this crystal clear. This is the #1 rule. Diphtheria is a clinical diagnosis first. You see that suspicious pseudomembrane? You don't wait. It can block an airway in no time. So you start treatment with antitoxin immediately. The lab work, that's for confirmation, not for making that initial life-saving call.
All right, let's dive in. Here's our roadmap for today. First, we'll walk through the entire lab workflow using a really simple mnemonic. Then, we'll figure out how to prove the bacteria is actually the toxic kind. After that, we'll look at infection control, super important. And finally, we'll wrap up with our ultimate weapon against diphtheria, vaccination. When it comes to lab diagnosis, especially for your exams, you need a system, a reliable framework. So we're going to use the mnemonic SSCIT. It'll help you break down the entire procedure into 5 easy, unforgettable steps.
So what does SSCIT stand for? It's simple. Specimen collection, smear and microscopy, culture, identification, and the grand finale, toxin demonstration. If you follow this sequence, you'll have the complete diagnostic picture every single time. First up, specimen collection. You're going to get a piece of that pseudomembrane and, critically, 2 throat swabs. Now, your examiner will almost certainly ask you, why 2 swabs? And here's your easy point. One is for the direct smear, so you can get a quick look under the microscope, and the second one is for culture. Simple as that. And here, we can really see why using the right stain is so important. Look at the left. The gram stain gives us our first big clue. We've got gram-positive bacilli in that classic Chinese letter arrangement. But then, look to the right. Wow, Albert's stain is the real star of the show. It makes the diagnosis so much more specific, staining the bacteria green while making those bluish-black metachromatic granules at the ends just pop.
These granules are a huge identifying feature, and, trust me, a favorite exam topic. You might also see them called Babe's Ernst bodies or Volatin granules. Think of them as the bacteria's little energy reserves, made of polymetaphosphates. And remember the special stains that make them light up? Alberts, Neissers, and Ponders.
All right, now for culture. You need to know 2 main types of media, and they each have a very specific job. On the left, you've got Loeffler's serum slope. It's all about speed. It's an enriched medium, so you get growth in just 6 to 8 hours, and it's fantastic for showing off those metachromatic granules. Then on the right, we have potassium tellurite agar, or PTA. This one's the specialist. It's selective, and C. diphtheria creates these classic black colonies on it after about 48 hours by reducing the tellurite.
So the main takeaway here is really simple. You pick your media based on what you need to do. Need a fast result and want to see the morphology clearly? You go for Loeffler's. Need to pull the bacteria out of a contaminated sample, like a throat swab with tons of other bugs? Then you use potassium tellurite agar to shut down all that normal flora. Okay, so we've grown the bacteria, we're done, right? Not even close. And this is a critical point. Just finding C. diphtheria is not enough to diagnose diphtheria. The disease is caused by the toxin, and not all strains of the bacteria actually make it. So we have to prove that the specific bug we found is toxigenic. The classic gold standard method for doing this is the Elek gel precipitation test. It's such an elegant little in vitro test where we can literally see the reaction between the diphtheria toxin and a specific antitoxin right there in an agar gel. Now here is the clever part. You take a filter paper strip that's been soaked in diphtheria antitoxin and you lay it down the middle of an agar plate. Then you streak your test bacteria at a right angle to that strip. If your bacteria produces the toxin, it diffuses out from the streak, while the antitoxin diffuses out from the paper. And right where they meet in the perfect concentration, poof, you get a visible line of precipitation. It's fantastic. Okay, this table, this is pure high-yield exam material. Seriously. Because Elek's test isn't just a yes or no for the toxin. It's a powerful tool for epidemiology during an outbreak. You can streak isolates from different patients and see how their precipitation lines interact. If the lines fuse together perfectly, the strains are identical. If you see a little spur, they're partially related. And if the lines just cross right over each other, the strains are unrelated. Now, while Elek's test is a classic, modern labs have faster options. The most common one today is PCR, to directly look for the tox gene itself. You can also use methods like ELISA to detect the actual toxin protein. And a key point for your exams, the old-school in vivo tests that used guinea pigs, they're considered obsolete now.
So once a case is confirmed, the mission changes completely. It's all about containment. We absolutely have to prevent the disease from spreading to anyone else, and that means specific infection control and prophylactic measures.
This strategy here has multiple layers of defense. First, you put the patient in isolation under droplet precautions. Then, for all their close contacts, think family, classmates, we launch this really powerful two-pronged attack. We give them a booster dose of the vaccine to immediately ramp up their immunity, and we start them on a course of antibiotics, like penicillin or erythromycin, to wipe out any potential bacteria before it can cause an infection.
And that brings us to the single most effective tool we have for preventing this disease from ever starting: vaccination. This has been one of the biggest public health victories in all of history.
These are your must-know facts. The diphtheria vaccine is a toxoid. That means it's the inactivated toxin, not the whole bacterium. It's made from a specific strain called Park-William-8. The whole point is to train our bodies to make protective antitoxin antibodies. Its potency is measured in LF units, and a lume is added as an adjuvant to give the immune response an extra kick. And here it is, the complete vaccination schedule you need to know for your exams. The primary shots are given in infancy at 6, 10, and 14 weeks as part of the pentavalent vaccine. Then come the critical DPT boosters at 16 to 24 months and again at 5 years. And it doesn't stop there. Tdap boosters are given at 10 and 16 years to keep that immunity strong through the teen years.
And this brings us to our final and really thought-provoking question. For decades, we had diphtheria on the ropes thanks to vaccination. So why are we now seeing outbreaks and a global resurgence? This really forces us to think about huge public health challenges like gaps in vaccine coverage, the problem of immunity wearing off in adults, and the absolute necessity of lifelong booster doses to keep our shield up against this deadly disease.